Hi, my name is Jales Fonseca. I am a PhD candidate in the sorghum breeding and genetics program in the Department of Soil and Crop Science at Texas A&M University. And today I'll be talking a little bit about analysis of diallele crosses. I'll present a brief review on the theory, and I'll spend more time showing some packages that are available in R for you to perform the analysis of Diallele. But before, thank you for the invitation. It's a pleasure to participate at the JR Biotek workshop this semester. And I hope this is interesting and can help you guys somehow. So to start, let's start with the definition. What is a diallele? A diallele is a diallele cross is a set of matings between genotypes. And the reason we develop those mating designs is to assess some genetic information about a specific population. So if you want to assess the general combining ability of a parent, of some parents, or the specific combining ability of hybrids, or if you want to assess heterosis and measure the magnitudes of some of those parameters, we can use diallele crosses. And based on the number of parents and the number of crosses that those parents participate, we can classify diallele differently. So for example, if we use all of the parents, we can develop a full diallele, or a half diallele, or a partial diallele. And I want to spend more time actually talking about the different ways for you to analyze diallele crosses. And as reported by Griffin, you can either assume your effects are fixed or your effects are random. So based on those assumptions, there are different methods for you to analyze your data and for you to assess the significance of some effects. And for you to do that, you can either use ANOVA, and when you use ANOVA, you just equates the mean square with the expectation of mean squares. I'll be covering in the R codes, and it's going to be way simpler to understand. And you can use linear models and use the ordinary least square method, or you can either use linear mixed models, and those last two, the linear mixed models and Bayesian regressions, are for those cases that you assume your effects as random. As I said, there are several packages in R, and I'll be sharing and using some of them throughout the lecture. So a full diallele, as I said, involves all the possible crosses among parents in a trial. So for example, here we have 5 parents, and this is the formula for you to calculate the amount of crosses you will get in a full diallele. So those 25 genotypes, those 25 crosses here, will include parents, which will be represented here in the diagonal, a set of F1s, which will be involved in here, the upper part of this array, and the reciprocals will be displayed underneath here. So for example, here is the cross with parent 1 and parent 2. And the reciprocal cross will be here with the parent 2 with parent 1. Okay, so we can also use a diallele without the parents. And this is the formula for you to calculate how many crosses or how many genotypes you will have after making those crosses. So among this, you will have 20 genotypes that includes a set of parental F1 here and the reciprocals. As I said, there will be no parents involved. A half diallele, you will only account for parents and a set of parentals F1. And the formula, again, displayed here. And when you have a half diallele without parents, you will only account, in this case, for 10 crosses. Okay, and partial diallele is just a reduced in a way that some people says that it is a more efficient way because you don't need enough space for you to test all of the possible crosses. And so this is the formula. And in this case here, you would generate 10 crosses if you have 5 parents. But okay, so now we know that there are several different diallele mating schemes. And what I want to emphasize here is you can use any of those diallele to extract information, but you need to decide what kind of information you want to extract. And according to the literature, there are several ways already, classical ways for you to analyze that kind of data. You can go to the Heyman's paper or to the Griffin or Gardner and Eberhardt, and each one of these um, 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 models or ways to analyze your data will give you different information. So in this case here, you would give you the general combining ability of parents, the specific combining ability among those combinations of parents, and also would partition the reciprocal crosses in reciprocal GCAs and reciprocal SCAs. But if you don't care about partitioning the reciprocal crosses or the reciprocal effects, you can use just Griffin, and that partition won't be generated if you use this method. But let's say you're more concerned about heterosis. So there is this model here presented by Gardner and Eberhardt, and that assesses heterosis and all of the nuances that can control heterosis. And in R, everything is already developed. So you just need to understand how to feed your data in. And I pointed out that these models here, they were developed with the concept of ANOVA, of using analysis of variances. And recently, there are more general ways for you to analyze that data. Instead of developing the specific sum of squares for each effect, you can go for more flexible models. And these flexible models can generalize diallels into a single framework. And that requires you just to make some reparametrization of the effects using some matrices. And those matrices account for these different types of analysis. And for those cases, we use linear regressions, mixed models, or Bayesian regressions. So I'm not going to give a lot of details, and I don't want to get into the heavy part of the theory, but I pointed out here that Griffin in 1956 already mentioned the importance of deciding if your effects are fixed or if your effects are random. And so this is a fixed effect, and this is a random effect. And this is how you calculate the sum of squares for general combining ability, the specific combining ability, and the reciprocal effects for the method 1, which is a full diallele. So now the parts that you guys are more interested in will start to appear. So here is how would you calculate the sum of squares for you to calculate the mean square of those effects. So here is how the formula was presented in the paper. And I want you guys to see that in the code that I'll be providing after this workshop, you guys can literally just go and check it is the same thing here. For example, the sum of squares for GCA is this quantity here, which is expressed here, minus this quantity here, which is this quantity here. So it will make more sense in our coding when we are running those things. But I'm just want to emphasize that is this first example that I'll be showing you will be literally the model that Griffin presented in 1956. The same thing for specific combining abilities and the reciprocal. So again, in the paper, you see all of this, a lot of formulas and how you can extract the magnitudes for the variance of the mean, the variance of the GCA, and everything is already here in this code that I'm sharing. And I hope you guys have a little bit of background in R, and I think it's time for us to shift gears and go to the analysis. Okay, so again, this is my contact. If you guys wanna reach me by email, please do not hesitate. If you guys have any questions, just send me an email, and I'll do my best to try to respond them. Okay, so as I presented before, a full Diallele, we will be using a package called plant breeding. So if you guys don't have the plant breeding package, just run this code without the hashtag here. And then for you to call the package, you just use this function library. And then, so now basically after you download your function or your package, you need to call this package to your R. And now it is available for you to start using some of the functions that are built in that package. So if you're having a problem to download this package, you can access the this website here that can give you more instructions of how to download. Sometimes you need to write require. So just it is here. So just Google it or read through it. And here are some of the codes that I'll be presenting. So since we are using this plant breeding package, they already offered you some data. So I will not be calling the data in from the computer. The way we're doing it, it's just calling the data with this function. There is a data in the plant breeding package. So once you use this function, it pops up here. So you can just click here and see what is this data about. So we have a full Diallele, we have males, females, and reps here. And then we have yield. Okay, so the formula that I presented there, it's basically this thing here. So once you run it, you will get all of those estimates. All of those formulas were already calculated. You just need to run this function here. And this is just an object that I can assess some specific outcomes using this sign here. So within this object that stores all of the data that was processed using this function Diallele1, I can just come here and see, well, what is the ANOVA? So the ANOVA for this trial is, we have 64 progenies because degrees of freedom is n-1. We have 4 replications, so it looks okay to me here. So sorry, this Diallele is about 8 by 8 for a grain yield, and it was presented in Sink and Chowdhury, 1979. So all of the codes here have a specification in front of it. So and I'll be sharing this after. So don't worry if things are not very clear at the first sight. So again, if you want to extract the effects, so the analysis of variances for GCAs and SCAs, and this is, again, we're considering this first example as a fixed effect. So if you see here, we see that GCAs are significant, sorry, and SCAs are significant. So you can keep extracting. So these are the estimates of each parent. So this is the general combining ability of parent 1, parent 2, so parent 3. So we're talking about yield, and this value here is very high. So this parent here is the best parent. So for example, if you want to produce a population with higher yielding potential, this parent must be in the cross. This parent here is also a good one. So this extract the GCAs for each parent. And you can extract the SCA effects, so the specific combining abilities of each combination of parents here. So we have a matrix full of those effects, and we can identify those best particular hybrids, not a hybrid, but those particular parental combination, yeah. And this is the reciprocal effect, and this is just all of those values that I showed in the slide, telling you the standard errors and the variance estimates for you to compare across each GCA. So if you understand, that's one thing I put it here. So all of the analysis I performed in here using this function that is in this package here, I broke it down, and well, I just put it here. The person that made it and made it available is Umesh, Dr. Umesh, and it's here in this website. So you can he allowed us to see how he developed the codes. And again, if you want to literally understand what's going on behind of that function, you can just click here, run it, and then come, and you will see that the ANOVA that was performed using this function, it's here. And this is how you perform an ANOVA using a LM function. And now for you to start developing the GCAs and SCAs and the reciprocals, you need to convert your data in such a way that your data is in a table like this. So remember when I was showing the different diallels? So this is kind of that matrix, that array, and the parent 1 with the parent 2, it's expressed over here. So that's kind of the similar idea. And then you keep running, and you will get all the values that were already performed before. So here is just for you to know how many individuals or how many parents are in your trial. And here is that quantity that I showed you on the slide. Here is the formula for you to calculate the sum of squares of general combining abilities. This for specific combining abilities. This is for the reciprocal crosses. And you keep going, and this is how you calculate the mean square error of your trial. So here we have 4 reps. So it tells you that for you to calculate your mean square error when you're using ANOVA and you have 4 reps, you just need to divide it by the number of reps. So again, you keep going, and then you will end up with that table that shows you the GCAs and SCAs. These are all the quantities that you need to develop in order to have the significance of those effects. So this is all of that function. That function does it all for you. You just need to run that. I just want to emphasize, see, I just want to emphasize that coders or people that work in developing those packages, they build those functions and they allow you to just fill up the data. So now that I finished, I want to show you how to fill up the data. Here in the data frame, you will put, like, for example, if you have a data set where your trial is, so here is where you're going to put it. So as I showed you before, I'm sorry, it is taking out here. So here is how you would put your data. This is the format to feed the data into the package. So the male, you would put male or parent one. If instead of a male, you have a parent one here. So this is how you call the column of different effects. So female, progeny is just the hybrid. It is the parental combination. And replications here is rep, and then yield is the trait that we are evaluating. Okay, so understanding all this allows us to go and present a different package now. Oh, sorry. This is those quantities that we ran already, just to construct all of those values that I showed in the slides. Okay, so a different package that you can use called LMDialer. And different from what I presented, that is a function that considers the ANOVA, and every time that you are using ANOVA, you need to equate the mean square to the expected mean square. And when you use linear regression, you can just reparameterize those effects, and you can extract the same kind of information. And again, this is a different package, and I would recommend you, so again, to install, you go to libraries, use this function, and then this code here. And Dr. Onofre is the professor who developed this LMDialer, and it is a pretty good one. It has very there's a lot of different models that you can run using this function, and you can either use lm.diallele, which is a function built in to the LMDialer, or you can use the LM function to run your codes. So here I'm just giving you an example of how to feed the data in. Again, using the same data that we were playing with, instead of putting yield down there, so now we're adjusting a linear mix model. Oh, sorry, just a linear model. And here you have your yield, and this is the sign here tells you yield equals, and here is where you create your parameters. So if you're using Griffin, the same model that we were using, we are trying to extract the GCAs, the SCAs, and the reciprocal, sorry, the reciprocal effect. So you run here, and you're gonna get the same values before. So 7 parents, you have 28 SCAs, you have the same significance and everything. So it is just another way for you to run your data. Okay, now we're gonna change the data. And we are now using a data that was presented in Heyman 1954, which measures the flowering time in Nicotiana rustica. And we will be using the LMDialer function. So again, you run this, and same thing. In this case, we have a different, so we add the block effect, and then Heyman is concerned about partitioning the reciprocal effects. So you can see that there are more effects in this model here. And then you will see the you will get GCAs, SCAs, and then the reciprocal GCAs, and then the reciprocal SCAs. To make easier to understand the differences between the models we saw before, here, now let me run this again. Here, for the Heyman model, partitions the reciprocal in reciprocal GCA and reciprocal SCA. And here we combine them two. So that is the difference. So it is more informative. Okay, keep going forward. If you are interested in studying heterosis, you can use a Diallele to assess the average heterosis for a particular variety, or you can measure the average heterosis of a specific variety in the cross. So again, if you want to understand more of the theory behind of these models, I strongly recommend you to read this paper. I really want you guys to know that there are some packages out there that makes your life easier to run this analysis. And again, so we will be using for the Gardner and Eberhardt model to assess heterosis, we will be using this data that is presented in Zhang 2005. And they just created a Diallele data set involving 5 lines. And you can see the data here. So parent 1, parent 2, block combination, then you have several environments, and then yield data. And because we have several environments, we need to partition our data. We need to consider environments within the data. So that's why we have all of the effects here, and then we have all of the interactions of those effects within the environment. And then you come up with all of the effects that the Gardner-Eberhardt model tells you or allows you to have. And then the same model I applied here without considering environments. And there is a debate, and there are some additional steps for you to consider environments on your analysis. And I might be recording. I can't record another video that consider the benefits of doing a 1-step analysis or 2-step analysis. It will depend on the questions. Okay, so another example here using a different data set. And this data set is from a Diallele experiment with 6 maize varieties and no reciprocals. So we'll be also using that model, Gardner and Eberhardt. And here we see all of the estimates. And one thing that I want to point out here is that in this case, we only had the data for the average across environments. So you need to include the the mean square error for the analysis, for the combined analysis, so you can extract the ANOVA without a problem. And you see that the significance, it's all here. So as I called the attention before, there is some additional attention. If you're performing your analysis in one step or two steps, you need to make sure that you're getting your effects tested right. So everything that I presented so far was based on the assumption of fixed effects. Now we're going to be discussing a random effect. And the random effect, instead of testing the general combining ability with the error term, you're going to be testing the general combining ability with the specific combining ability. And again, we're going to be using the same full Diallele that I presented first time, the first data set. Again, we can just come here and see it. So it is parent 1, yield data, parent 1, parent 2, rep. It's the same one that I presented before. And again, here you will have the ANOVA for the model 2. So this implies a different F-test, which is I just mentioned. And here is the variance components for model 2. As I said, if you guys are interested in understanding how the calculations were performed, they will be all described here. And for time's sake, I will just be running here and showing this first two. So these are the effects that were calculated in the model 2. So, and then you ended up with those estimates that were presented before here. It is exactly what we got using just the function. So I showed you guys already the plant breeding package and the LMDialer package. And those are great for estimating fixed effect. I showed you this random effect just to start talking about a really good package that a lot of people are using to perform Diallele analysis and even predictions using a mixed effect model. And the package called SOMER, and it was developed by Dr. Cohubias Pazaran. And you guys can read the paper and read the vignettes about this package. So again, when you're assuming a random variable, you're using the RAML estimates, which are the restricted maximum likelihood estimators that were first presented by Patterson and Thompson in 1971. I don't recommend you guys to read, but if you guys are interested in reading a little bit about stats, it's a great paper. So this function here just clean our environment here because we're using a different set of data. And again, to install a package, you just go install packages and then you put SOMER on quotation marks. And since I already have the package, I'm just gonna get the package to work in my R now. So library SOMER. And inside of that package, they also provide a data set called half Diallele. So which includes 7 parents. And how can you assess the genetic, the general combining ability of those 7 parents using this package, assuming the effects are random? You just run those line of codes to start preparing for the analysis. And then for you to develop the model, you need to use this function, MMER. and this is how you feed your data in. So sugar is the, did I show the data here? This is the trait that we are measuring. So this is the trait. Before it was yield, now this is sugar, and we have male and female in our crosses and reps 1 and 2. So how you field your data, how you put your data to work, this tells you that this is the trait that we are assessing. This number 1 tells you that there is no fixed effect but the mean. So this is how you write the fixed effect. So for example, if you have like environment, instead of 1 here, you would put environment here. So this is how the package was developed, and you need to follow the pattern. So fixed effects is just a mean, so you put a 1. So what are the random effects? This is female and male. So as I said, as a more generalized way for you to analyze Diallele cross, instead of using ANOVA and developing a mathematical procedure to calculate each effect, here they developed a set of matrices that allows you to just use this code, and they will overlap the matrices. It is a function that tells the column of male and female and have a perfect correlation of 1, and allows you to extract the GCA information from those parents. And then you can get the summary. So again, the summary is a function that summarizes the output. And you can see that all the parameters for you to assess how good is your model. So for example, if you're comparing this model to another model, you would measure the Akaike number, or the Bayesian information criteria, sorry. Akaike information criteria and Bayesian information criteria. And so here you will see that the variance component is this, and for you to extract that, this is the variance for the general combining ability of the parents. So if you want to extract the values of each parent, so again, use this function here, and this tells you which parent provide higher amount of sugar in a hybrid combination. So the parent number 3 here is the one that increases more the sugar content. So going forward here, we can also use some relationship matrix to help increase the accuracy of our predictions. So for example, remember when I was talking about a partial diallele, not all of the parents are crossed. So how could you increase information to predict the performance of untested parents? You would create a relationship matrix using either a pedigree information or a molecular SNP information. So here, as this package provides, it's just a fake covariance structure. But once you have DNA data, you can calculate the variance covariance matrix that allows you to increase the information among the parents. So in this case here, for example, when we run this code, what we're saying is none of these parents here are related, but parent 1 and parent 2. So they're let's say they were half sibs. So any information that comes from these parents here, you would increase information. You can extract that information to predict the performance or the GCA of untested parents. So depending of how good are those covariances, you can increase and maximize and optimize the estimations of your GCAs. And this is how you include that information in the model. You just create this variable here, GU equals A, which is the variance covariance matrix. Then you put geno because you need to put this data in a structure. So let's go back here to the data. Geno is just the combination between male and females, just a hybrid. Let's say the hybrid. Okay, and then the summary again. Oh, sorry. Let's run this. Yes. And then you see again, we have 7 parents, and the GCAs of the parent is this. And we can have some information from the geno, which would be the kind of the SEA. And I think, oh, there's another example here using a different data set, now using a full Diallele. And I used this data set here to tells you, to give you an explanation of, or an option to include more than one trait in your analysis. So you can either run this and you will come up with, this is, so as I said here, 36 winter bean hybrids. So you have 6 females and you have 6 males, and you can extract the variance for GCAs of females here, the variance of the GCAs of males here. And this is very similar to what we would have if you run a factorial 2. But for this case here, we would include the specific combining ability of female by male. And again, I will not be covering factorial designs because it is kind of a similar what we're seeing here. We would only include female by male. And now we would have another effect there, which will be the specific combining ability using a linear mixed model. Oh, sorry. Okay, so as I was saying, that data set allows you to have a multi-trait analysis or a multivariate model. So how we develop that kind of model? Again, we use the function MMER, and we use it's another function that combines multiple traits, and those multiple traits will be used to assess the general combining ability and the correlations among those traits. So again, here we're putting 1 because the mean is the only fixed effect in this model. Random effect, when we use a multivariate model, we need to create some other variance covariance structures to account for each of the trait. So this is how we specify those effects. And this is how we specify the error term. You can model the error term with heterogeneous error, but let's keep this simple. This is just the compound symmetry model. Compound symmetry model just tells you that the error term will be similar on the diagonal of the matrices that describe the error term of your model. So again, you have the summary of this model we'll give you the estimates, the mean for each trait, and also the combining abilities within each trait, and for the correlation between two traits. And how can you assess those covariance correlations? Using this code here. So the correlations within the females, the GCAs of the females, you have a almost 0.6 correlation between stem and pods, and a negative correlation with seeds, stems and seeds. Same thing for the male, and this is for the residuals. So I think there is a lot already for you guys to play with, but I'm happy to come back here and go over through more details. And a final remark that I would like to tell you guys: instead of the SOMER package, there is also another package called LME4 that allows you to assess factorial 2 models, but it is challenging for you to assess Diallele crosses. So I encourage you, if you want to use a random effect, I would encourage you to use SOMER. And there's also Bayesian approach. I don't think I have enough time to cover the Bayesian approach of Dialleles, but these two packages here are recommended. And for final thoughts, where can I find information about packages? Because this is for Dialleles, right? So sometimes you want to learn how to do principal component analysis. You want to use different packages. So this is where all the developers make available their packages and their codes. You can just Google them, Google the package, or just go in this website here and look for the package that you're trying to learn. And if you want to see how they develop it, all the developers, when they publish their research in R, their research using R, they are developing R packages, they need to open the source code. So if you want to see how they are developing all that, if you like this kind of thing, I encourage you to go to this website, GitHub, and you can see the source codes for all these packages that I presented and all the other packages that are available in R. And one thing that it is really helpful is sometimes those developers, they write vignettes, and those vignettes are just tutorials that teaches you how to use their package, how to use their functions, and how to better provide information from the package to users. And with that, I would like to point out some references that you guys want to read about to get more information. And I really appreciate your time. I hope that this was helpful somehow. My email is there, so please do not hesitate to contact me. I'll do my best to answer those emails. Thank you for the opportunity, and bye.